This is a production of Cornell University.
So I thought I'd start off with a little introductory slide. First of all, showing the world as it really is. You'll notice New Zealand is that little country to the left of that continent, Australia. And as the saying goes in New Zealand, Australians are okay. I mean, we as New Zealand, another Kiwi in the audience here. We always like to say that as New Zealanders, Australians are okay, just that you wouldn't want your best friend to marry one. So, but apart from that, so a little bit about me. I've been here at Cornell since 1994. I came from New Zealand, where I first started working with at a government research organization where it was privatized and all government research was separated into different institutes. That's a long story all by itself. In the middle of that time, I did my PhD at Rutgers. So I won't tell you what years those are, because there's so many in the room that won't have been born by that time. So my area of expertise is in post-harvest science, or post-harvest physiology, as it's sometimes called. And it's really an integration of the study of the ripening and senescence of horticultural products, and focusing really on maintaining the storage quality of those products in a very broad sense, as I will mention in a moment, as affected by pre-harvest and post-harvest factors. And if you want to sort of put a moniker on it, it's about reducing food loss. That's really what the work is all about. Oh, all right. So I've been working, or I work on just about any crop over the years, often tomato, strawberry, bulbs with Bill Miller, who I think spoke to you fairly recently, even played with a little bit of Arabidopsis. But most of the work that I have been focused on in the lab is on apples, mainly because it's the most important horticultural crop in New York. And really, we're at a sort of what I call it a time in history where there's an amazing transformation going on in the apple industry, both in terms of the variety mix and in terms of the storage technologies that we're using, and I'll touch on that in a little bit. So in the center, we have the apple, our work, which is ripening physiology, quality factors, susceptibility to storage disorders, which is a huge part of our work. And the driver for all the problems in the world, and I will show you a photo in a moment of how many disorders this apple gets. But that corner, that top corner, is the Honeycrisp. And the Honeycrisp apple has really revolutionized the apple industry, because the industry has finally realized that people are willing to pay more money for an apple they actually like. Many of you are familiar with the apple called Delicious. Washington State industry turned that into an aspiration rather than a reality. And we want to make sure that we don't do that with many of these new varieties. One next to Honeycrisp on the top is Gala. That's an apple from New Zealand. That is actually the number one apple in the US. A big transformation from Delicious to Gala. Delicious plantings are plummeting. Gala plantings are rising. And then we have another apple, Pink Lady, which is an Australian apple that is also relatively new on the market and is expanding very rapidly. But Honeycrisp has been the most interesting apple, as I mentioned. It's a very strange apple. The reason it stays crisp is it doesn't have the genes involved in fruit softening, or has very low expression. It also has very limited ethylene production, which in apples are autocatalytic, which means that they produce more and more ethylene as they ripen, and that's associated with softening of the apple and general deterioration of the quality. So the thing about Honeycrisp is it has been used extensively in breeding programs. So on the bottom, we have Wild Twist. We have Snapdragon, which is a breeding selection from Susan Brown at Geneva, and Evercrisp. Just before I move on, so interacting with those new cultivars, there are so many pre-harvest factors that influence the way these apples perform. Then our work has involved sensory work, nutrition work, and most recently working with Jenny Niffen and Connor Lane, a PhD student that we share, working on the microbiome. I mentioned Honeycrisp has some problems. This is actually just a few of the photos. It is a remarkable apple in terms of its susceptibility to just about every apple disorder that you can manage. And you would say, then why do people grow it? That's why the reason for that is it was earning about $1,000 a bin for growers. The context of that is a normal apple variety like delicious would be $200 a bin. So a lot of orchardists have really paid off the farm with those investments. Our primary research priorities, I'm just going to go through these very quickly. In the 1996, North Carolina, Sisler and Blankenship patented 1-methylcyclopropene as an inhibitor of ethylene action. There's a long story here as well, but essentially it became certified or allowable for food use in about 2002, and it has been a remarkably exciting ride to be on this the MCP bandwagon as it has expanded throughout the world. And what's been really cool about it is, or what is interesting about it is, it's a treatment that is absolutely perfect for apple because it keeps the apple from ripening, but it has many other problems like delaying or inhibiting ripening rather than delaying ripening for many other apples. Dynamic controlled atmosphere is a new technology that is where we're able now to monitor the stress in an apple as you lower the oxygen concentrations. On the left-hand side, that's just one example. That's a harvest watch system based on fluorescence. And sort of a simple diagram on the right-hand side. Here's your fluorescence signal. As your oxygen concentration comes down, the fluorescence will tell you that the apple is stressing, and as a result, would die from anaerobic respiration. But we're able to bring up the oxygen level to prevent that, and that's a major technology that we're working on at the moment. Did you put your finger up? How long do I have? 1 minute. All right. Well, then I'm definitely going to zip through. I would just say projection of bitter pit. On this area, you'll see all these scatters. This is showing the relationship between minerals and bitter pit, generally very noisy. In our lab, we've developed a passive method, which the industry is using now to predict the amount of bitter pit and what they should do. So I'm just going to finish off then just with an update, sort of, and tell you the sort of funding that we have received. It's largely being specialty crop research initiative funding. The one on the left top is the one that we have currently. We're about halfway through, and that's developing dynamic controlled atmosphere storage, safe use of dynamic controlled atmosphere storage throughout the US. And then on the right-hand side, recognizing many of the faculty that have been partnering with me. I'll stop now. So I didn't think I didn't think I was that slow, but however, it is what it is. Do I have time for questions or no? Oh yeah.
Any questions for Chris?
Yeah. What do we measure the fluorescence on? The skin of the
What molecules fluorescent?
is the chlorophyll signal. It's fluorescing. It's from the skin. Yes. Yeah, I know.
So if you have to do this because you cannot just monitor oxygen by itself, you need to also just see the reaction of the apple. Because I'm wondering why not just look at oxygen in the atmosphere.
So you're talking about this
Yes.
slide here, yeah. So this is just a sort of an experimental one.
Okay.
So what happens in the real world is the oxygen levels are brought down slowly over time within the commercial storages. They take it down until they see that signal, and then come up 0.2% oxygen, and then you check that every month or so. And there are different systems where all the information is going from the iCloud to a central place, which will help tell you what you should be doing. Yep. All right, thank you.
Well, hello. Well, thank you, Plant Biology, for inviting me. I am going to talk about our quest to harness fungal innate immunity responses to bacteria in order to control fungal pathogens of plants and animals. And our model system is actually a post-harvest pathogen in mucormycete molds. These are early divergent fungi. They are ubiquitous soil saprotrophs. They happen to be post-harvest pathogens. They are devastating opportunistic pathogens of humans. They have some practical applications like production of polyunsaturated fatty acids. And they are also very poorly understood, very low studied. And I'm sure you are familiar with these organisms. So in the course of our studies, we discovered then that when these fungi are faced with bacteria that don't produce secondary metabolites harmful to the fungus, but the fungus perceives these bacteria as antagonistic, it will respond with activities that resemble innate immunity defenses of plants and animals. Plants and animals have evolved innate immunity systems convergently, and we are trying to convince everybody that fungi have innate immunity systems as well. So what happens? Fungi remodel their cell walls like plants faced with pathogenic microbes. They produce reactive oxygen species. And recently we discovered that fungi die suicidal death in response to pathogens, like hypersensitive response in plants or pyroptosis in animals. And just to walk you through our experimental setup, this is our model fungus, Rhizopus microsporus, a colony growing on agar facing this otherwise friendly bacterium that the fungus thinks it's an antagonist. And I hope you can appreciate that there is a little bit of a zone of inhibition. So once we realized that these fungi actually die, regulated cell death, immune death, we became interested in what kind of death it is and how it happens and everything that surrounds it. And because it is difficult to study death within intact colonies, we switched to working with germinating spores. And from other fungi, we knew that adenylate cyclases can sense bacterial peptidoglycan. So we thought, maybe adenylate cyclases of this fungus, Rhizopus microsporus, and it has two different copies of these genes. Maybe they are also involved in sensing bacteria. And so we created disruption mutants in both of these genes, and we assayed them for life or death. And this staining is done using live dead staining. Live cells are green, dead cells are red. These are spores germinating alone, spores germinating in the presence of this supposedly friendly bacterium. I should say, and they are dying, and I should say that the rate of death is dependent on the bacterial titer. The adenylate cyclase 1 mutant when it's exposed to bacteria, does not die. So we think that this gene is a positive regulator of death. Adenylate cyclase 2 mutant dies exposed to bacteria, dies at a much higher rate. So we think this gene is a suppressor of death. So adenylate cyclases sense peptidoglycan. Peptidoglycan is a component of bacterial cell envelopes, and it is also a microbe-associated molecular pattern. Plant pathologists amongst us know about MUMPs. So this made us think about MUMPs in general. And in addition to peptidoglycan that is inducing immune responses in plants and animals, there is also lipopolysaccharide, lipotechoic acid, which are components of bacterial cell envelope, and flagellin, that is a component of flagella. So we started doing these experiments, exposing fungal mycelia, not just to bacterial cells, but also isolated MUMPs. And hopefully I can, and this was the very same setup that I showed you already. And hopefully you can appreciate that when bacterial colonies are exposed to peptidoglycan here, they are inhibited as well, so that peptidoglycan elicit a very similar response to live bacterial cells. And this is work of a graduate student in the lab, Dilia Tota. Dilia took this challenge seriously, and moving back to our spore germination system, germinated spores in the presence of various MUMPs. And this is the product of last 2 weeks of experiments, so expect more. Dilia found out that lipopolysaccharide and lipotechoic acid cause immune death in germlings exposed to them. And these are microscopy images of these particular interactions. In this particular staining, dead cells are still red. The blue are the nuclei. So this is somewhat different principle. So I hope that with this, I convinced you that bacterial MUMPs induce regulated cell death in fungi, and exposing fungi to bacterial MUMPs presents a potentially promising strategy to control pathogens of plants and humans. And with this, I would like to acknowledge people who did all the work. Dilia, Maria Laura Gaspar, and Carlos Lax generated the data that I was presenting here, but this work rests on shoulders of giants, current and past members of the lab. And before I finish, I would like to say that if you want to learn about symbiotic associations, I teach a course this semester, and we talk about not only antagonistic interactions that I just talked about now, but also about mutualisms, because this is what we are actually interested in. Thank you.
Simple question, but I don't know if the answer is simple. What's the difference between regulated cell death and programmed cell death?
That regulated is a little maybe newer term that they use in the literature.
Okay, so it's being a shift in the language.
Yes. Yeah.
Simple question. What is the benefit
Of death?
of that? Right?
Oh, listen. The benefit of death is containment of microbes. So microbes that have penetrated cells are taken down with the dying cell. But there are also other benefits. For example, dying cells release damage-associated molecular patterns, and these damage-associated molecular patterns trigger and proliferate the response. So there is also a signaling to neighboring cells. And in case of fungi, we think that this is signaling to the spores cells in the population.
Do you think these MAMs are generalized signals of bacterial presence?
Yes. So both plants and fungi, plants and animals respond to those.
Specificity in terms
No.
of which?
No. There is a threshold, but no specificity. Thank you.
Thank you, Teresa. Hi, everyone. Thank you for the invite, first of all. And a little disclaimer, I've given this lightning talk twice in the past 3 months. So if you've seen it already, I'm sorry for the repetition. And the second thing is, I have a background in molecular plant pathogen interactions, work with plant breeders, but now I work in the intersection of social science and plant breeding. So any students who want to talk about a very non-circuitous way of getting to your career goals, please talk to me. Okay, so I'm going to talk about what we call the Equal Lab. So this is our research group in plant breeding. Because I don't have a whole semester of teaching opportunity, I'm going to start with the bottom line, and that's to say that we know from decades of research now in globally, gender inequalities are pervasive in agri-food systems. We can very confidently make this point. There's a lot of data that points to this. So this is our departure point for all the work that we do. So I'm going to start with the bottom line. What this means in practice is a lot of scholars, this is a recent review by Jemima Nijuki, talk about how components of agri-food systems, from value chains to food environments to consumer behavior, are all affected by systematic gender inequalities, right? Again, this is based on a lot of work talking about how women's agency is shaped by social norms and how then they can engage in different parts of the food system. There's been a lot of work on women's access to and control over resources in agri-food systems, in farming especially, tons of work there. How does that shape how they interact, and so on and so forth. All the way to kind of more macroeconomical institutional factors that dictate those social norms and engagement in food systems. So what does this mean? More recent thinking looks at what's called gender transformative change. What that means is we want to open up more spaces for women and other marginalized identities to engage in agri-food systems. Okay, that requires social change or change. So if you look at the literature, you see that change can happen at different steps. It can happen at individual level by gaining agency. It can happen at the household level, looking at inter-household relations. It can happen at the community level, who's allowed to do what is a norm in communities, organizational and microenvironment. In parallel, more recent thinking in agricultural innovations tells us that you cannot innovate detached from social context. Okay, when you're making an innovation, when you're conceptualizing of a new agricultural innovation, you must take into account the context that you're innovating for. And this, as I see it, is where we are. Our lab kind of works in this space, okay? And what we try to do as the Equal Equitable Agricultural Research Lab is look at the intersection of kind of gender transformative gender research and how we can have context-dependent innovation. In our case, that's crop improvement or plant breeding. Okay, those are the innovations that we hone in on. We try to do what we do differently. We borrow from a lot of really interesting examples of how to run a lab, how to do research together, how to interact with one another. So we really espouse feminist research practice. We do almost all inter or transdisciplinary work. We don't really focus on single disciplines. And all of our research is international. We don't have a lot of local research, so a lot of collaboration is involved. And we really focus on our core values as a lab and how we interact with each other through those. We're not wedded to any crop or context, so we're kind of agnostic because you'll see a lot of the replication of the same issues are global, right? Crop and context don't matter that much. So these are kind of the suite of crops we work on across different, projects and the different countries we work in, a lot in Sub-Saharan Africa, more recently South Asia, and even the US and Central America. So you're probably thinking, what does that actually look like? What does she do? What does this lab do? So I wanted to give some examples. So at the individual level, some of the earliest work that we did over the last decade is looking at trait preferences. So when you're a breeder, you really care about which traits are important for whom, how do I target those, how do I kind of bring those into my breeding program. We've done extensive social research in cassava in Nigeria specifically, but also other countries, to try to get beyond farmers want X, right? There's no one group of farmers. There's no one farmer and one trait that everybody wants. So we try to break down social differences. We look at region, we look at gender, we look at poverty level, food insecurity. The whole idea of this is, these are kind of the intensity of preference for traits. We want to hand over more nuanced information to the breeding program to say, if you want to breed for food insecure women in the Southwest, these are the suite of traits you need to care about, right? Really social context coming out strong. And there's been a lot of variation on this kind of work that we've done. More recent work that I'm really interested in is what's called intra-household research. Economists, feminist economists really engage in this literature. That means within a farming household, how do people interact with one another? A really interesting work. We're bringing that to crop improvement or plant breeding. What you see here is kind of a kernel density plot looking at fingerprinting data that we take from fields of farm households. And we have self-reported adoption rates of cassava. What we're trying to look at is who knows what's being grown where, how, and why, okay? And what initially you see is who is more accurate, quote unquote, can be misleading because depending on who you ask and how, you get very different adoption rates, which makes sense, right? You're asking the head of the household, they may not know about some of the plots. If you go to the plot, someone may know more. So this is a part of our advocacy to say, if you really care about adoption metrics for plant breeding, you need to not just take one person from one household to tell you what they're growing, right? Unpack that box. Okay. More recent work we're also doing is community level. This is a seed systems project. If you're interested, I can talk more about it. It's called Muhogo Bora. We're entering our second, third year phase. This is really fun. It's all in Tanzania for cassava seed systems. What we do is we match social interventions with seed system interventions. So we go through school-based interventions, nutrition training, but also training around gender-based violence, land rights. How do you kind of not just provision seed, but provision it in a way that'll lead to social change? So really fun work we're doing around this, if you're interested. And penultimately, we look at organizational. We have 2 papers coming out this year looking at conceptualization of what it means to be a leader in international ag. How is that conceptualized? There's a lot of work on that being very masculinized, for example. Who is a good leader is really biased. But we also look at care responsibilities. Who's doing care work in academia for plant breeding, for example? So pulling these out to look for organizational and institutional change. And lastly, this is a paper we published last year looking at kind of broadly doing a scoping review on how trait preferences are collected at the global level over 50 years to pull out gender geography and donor bias in those data and how that shapes the information we have as a community. And I think with that, I'll just end. These are our lab members. We work a lot with different reading programs, and those are our funding sources. That's a QR code for the lab. Thank you.
So the characteristics, justice, and some other things there, one of them was humility. Can you, I mean, how do you define something like that?
Those actually came out of a lab retreat where we kind of played with different words that really describe. And I think the root of it, I can't, kind of judge who the people saying that understood. But what I understand of it is, if you go to work with a breeding program anywhere in the world, you have to walk in assuming you know nothing, right? We can't come across as being like, oh, I know all about this. So we always enter with humility, right? Teach me, tell me what you want. How do we work together? So it's always a point of kind of, I'm coming in to learn from you. And that's how I understand it, because those relationships are very important. So to cultivate those well.
Very broad question. Cornell's undergraduates now are about 60% female. And I think that's probably not unusual. It's getting close to 2/3. You can comments on that and how it very broad.
I can talk all day about that. There's so much literature on what's called the leaky pipeline, the sieve, the barriers. You can look at all the numbers globally. Those numbers peter out. They cut off. If you look at higher management, full professors at Cornell, it's 70% men and 70% white. So those numbers always end up dropping off as career advances. And some of the things we're looking at, like management leadership and care responsibilities, invariably shape those experiences. More women drop out of careers because of conflict with care responsibilities and people assuming that the woman should be the one doing caring. There's very complex social things happening. But I can very confidently say that does not hold. We've had enough generations now that those numbers should have trickled up, and they haven't.
How do we start to think about these cultural contexts in other fields? I think it's difficult when we're working in a lab to kind of make those connections. Do you have any?
Yes, I do. So we engage a lot with what's called science and technology studies. There's a whole department on campus. If you're interested, they study the sociology of science. And a lot of that work has happened in labs. So Barbara McClintock has been actually a subject of study, which is really interesting. There's books written about it. How people conceptualize science is shaped by their social identity. So whether you're in a lab or in a field or you're doing this work, there's always a thread, right? So understanding those social dynamics doesn't matter where you are. Could be in a lab, things could be happening that mimic very much what we're seeing.
Very much. All right, thank you very much for the invitation. There's another great opportunity to share the work that we are doing. So I want to present here the Farmers Data Lab, which is a research and extension program. And we called it this way because I'm building on the experience that I have in precision agriculture with all the equipments, the software, the analytics, the data handling. And what we are trying to do is to take this and put it to the service of farmers who are experimenting in their field. So it becomes their data lab, basically. Okay, so we are kind of building, trying to respond maybe at some of what we are seeing globally in research. So I put the entire quote here. From a very young age, we are taught to break apart problems to fragment the world. This apparently makes complex tasks and subjects more manageable, but we pay a hidden enormous price. And the price is kind of when we try to put it back together. And the analogy is a broken mirror that you try to put together. It's never going to look like the mirror you had before it was broken. When we give up this illusion, we can then build learning organizations where people continually expand their capacity to create the results they truly desire. And I was reading this in the book, The Fifth Discipline, and the learning organization, I liked it because to me, that was a way to kind of keep the mirror as it is, but you bring enough people together so that it becomes more of a systems approach or systems research. And so this came out last year, I think. But what this kind of little news clip was saying is that there are 70 plus organizations at the COP 28 that were asking for more food systems approach, meaning that kind of along those lines, stop breaking apart, doing those little things, but if we could have ways to learn with the system in place. We kind of think that it may be slower, but in the end, when we have a solution, we have a solution that works in the system. Okay, so we have this in mind when we try to build that Farmers Data Lab and to learn with the farmers. Okay, so what we are aiming to do is to bridge 3 disciplines together, okay? You see the first is agronomy. That's my background, and I have students that studied in agronomy. For consistent interpretation, we have the agronomy science that we can apply for soil and crop science. But then after that, we are working with data scientists because we think that bringing the data science to this, that will increase the certainty because now everything that's happening in the fields of the farmers, if we have the data to document it, then we have a much better opportunity to have a conversation around those data and to really understand each other without just it being anecdotes or conversations and impressions. Okay, so now we have the data to back it up. And then coming right after Hale, this is great. We are integrating social science, which we think is an inherent part of this systems approach, because if we're not able to work with the farmers where they are, then it will be very difficult to be effective and to create value. And so the approach that we have here with social science is that we are always after, okay, what are you trying to do with your experiment? Okay, what are the pains that you have to reach there, and what are the gains that you get if you succeed. See, so with this in mind, no farm is the same, but we are able to kind of have a more effective approach. Okay, so I've mentioned the word experimentation on the farm. So this OFE stands for on-farm experimentation, and this is the target of our operations, okay? We're not going to work with all the data of all the farms because this is kind of all over the place and it could be misleading. What you see somewhere maybe if you don't have the entire context, there are so many factors that it may be impossible to interpret. However, when the farmers experiment something, now they try, they have either a different rate, a new approach, like they will see they tweak their machinery, or they will try a new variety, or they will try something either side by side, just a strip, or 2 adjacent fields. I mean, they have their own ways to do it, and they use the landscape to learn. When they are doing that experimentation, this is where we can capture the data, and that becomes very a great opportunity to learn and to try to help them learn more from what they get, but also bring it into a bigger picture where others can exchange and everybody can learn from this. Okay? So concentrating on farm experimentations where the farmers lead the exploration. Okay? We're not telling them what to do and how to do it at all. Basically, they would do it if we are not there. See? It's just that we come in and then we try to enhance that process. So this is a systems approach because what they are doing in that specific season, that specific field, it aligns with their economic context, their soils, the type of crops they are doing, the machinery that they have, the human resources, the capacity, you see. So just because of this, because when they become the leader, it becomes kind of systemic because what they are doing it kind of makes sense for their world. We work with real world data, and this is on one side awesome because now we learn from the real world, but on the other side, a total mess. But the third part is that this is change in progress. Okay? We want farmers to go from conventional or more extractive agriculture to sustainable agriculture. This means we need to have a lot of change going on. Okay, and this is how they will change, by experimenting with new things. And what we think is that if we are there, then we can improve and serve this process, basically. Okay, so to do this, we want to provide expertise. Okay, 1 minute. So things that farmers may not have access. For instance, they don't have time to go get the specific samples. They don't know how to process the samples. They don't have the time or skills to do all the data science in the background. Then we want to be there for them. We have resources. I talked to you about my lab in Precision Ag. We have drones. We have tools, softwares, and things like that we can put to their service. And then we are hoping that this will have several outcomes: validate better certainty for the experimentation, capture innovation that can serve to others, accelerate the change that we want to see towards sustainable ag. And the idea here is that we want to scale the learning process rather than scaling one solution that we may have developed in our lab and try to scale it to everybody. See, so that becomes a little bit of a learn how to fish rather than give a fish. Okay, I'm going quickly, trying to do that in 1 minute. Several things that we are doing along the lines of trying to make this vision a reality. Okay, so we are conducting campaigns. We don't have a clue what we are doing, but doing it for real. This is when we learn. So last year we worked with 24 farmers, but we have a new season coming with several new experiments. And while doing this, we learn a lot. We developed tools that kind of lubricate the system, because right now when we have to go there and write everything on a piece of paper, that's tricky. So now we learn, okay, let's find tools to help farmers give us their data so that we can organize it and then return it the best way. We are conducting surveys to learn what is the state of on-farm experimentation. And so far what we see is that up of 90% of farmers experiment year in, year out. So this is non-trivial. Then after that, we have a new website to kind of for the extension part where we exchange resources. And we are working also with international collaborators to kind of bring it to the forefront of science, where this is a scientific approach. So to do this, we need to have kind of a global effort. So we are working on the global on-farm experimentation network. That's it. Thank you very much.
I think this is really fascinating, and I really like the farmer-driven approach. Do you ever have to do anything to develop trust with farmers that you work with? 'Cause it seems like a lot of the times there isn't a lot of trust between farming and academia.
Yes, this is a great question. And this is something that we learned on the spot. So what we observe is that you go and ask who is doing some experimentation of their own. And then you get this many responses. And what we understood is that as scientists, we dismiss this as being an anecdotal process, not scientific. So for them, they are doing it year in, year out, but they would not share this with the scientists who would look down at their approach, you see. So we had actually to really work on, go around this and repeat that, yes, this is a valuable process before we can build that trust so that they are open and they understand what we're trying to do, you see. So yes, this is an important factor. And I mean, maybe to add a little bit to this is that I kind of emphasize that we will follow their way to do. And then with this, basically, we're not asking much from them besides kind of having an exchange. And what becomes trickier is then to convince scientists that this is science, you see. So it's kind of work on both sides. All right, thanks.
This has been a production of Cornell University, on the web at cornell.edu.